So this is my presentation on cloning and sequencing the GAPC-GAPDH glucose regulating gene from spinach into an E. coli plasmid. Spinach being Spinacia oleracea. This is from the biology department at Western Carolina University. So the introduction to this is that here we are cloning the glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. It's a genotype from a spinach or dandelion sample to be inserted into a plasmid, then cloned into Escherichia coli. So then we can scan the resulting sequences using Genius to find homologs. So the gene that we want to clone with PCR is the GAPC in spinach. And so the black timeline right here, if you come a little closer, you can see. So we start off with DNA extraction, then do initial PCR right here, then nested PCR right here. Then we ligate it into these cells, and then these cells are then transformed and amplified. Then we use plasmid purification, then restriction enzyme digest, which later on will be DNA sequenced by some laboratories. At that point, we will later use bioinformatics analysis. So this is actually created by, if you see this screen right here, it was created by the BioRad laboratories. This comes from the Explorer Cloning and Sequencing Explorer series. So this is actually a 332-page presentation that our wonderful professor, the TA, or Christopher Bayer at Western Carolina University, has gone through all of that for us. So thank you to him. But for the abstract of this, we discovered that the purpose of our cloning is to transgenic organisms. And these are the transgenic organisms. So we do this by first extracting the GAPC version of the glyceraldehyde 3-phosphate dehydrogenase from spinach. Then we elongate it using PCR, or polymerase chain reaction. Then we ligate the plasmid vector into E. coli, and then we clone the target transgenic E. coli organisms. So when they're cloned, they produce more of themselves, of course. The primers did not anneal strongly to the dandelion on the initial PCR right here. You can see dandelion, if you come a little closer, this is dandelion, and you can see that it didn't anneal very well. So instead we used spinach because the PCR annealed much stronger to that. The spinach bands were 1,000 base pairs long, and the length of the base pairs was found using gel electrophoresis. The purification ligation was used to ensure that contaminants were not ligated into the E. coli. Below, you can see a detailed view of the PJET 1.2 plasmid, which if you come a little closer, this right here is what the PJET 1.2 plasmid looks like. Very detailed view. So how we do this is we get the plasmid DNA and the competent E. coli cells. Then we add the PCR fragment into the plasmid right here, which is then creating the transformed cells, which you can see right here. You transfer it to the selective media, which you incubate overnight. And then you can pick your colonies, which we have then done. So you can actually see the results of this over here. This one only produced 2 colonies. 2 colonies, which is, it's not very good. But the P-gap, the spinach P-gap, actually did produce tremendous amount of colonies, and the P-gap was very successful. Keep in mind the P-gap is the control. So this is actually the one that we cloned. And although it's not as successful as the P-gap, it still worked. And since it did work, it was enough to be gene sequenced later on. So the initial PCR reactions of spinach created 1,000 base pairs. Dandelion was only 500 base pairs. Our Arabidopsis control was 500 base pairs, and the water was the negative control, so it created 0. Onto the nested PCR, the water band is just a primer, the primer dimers. You can see a few primer dimers in there, and don't worry about those. Those are just normal. Arabidopsis has a 1,000 base pair smear, which is up from the 500 base pairs in this one. So it's doubled using nested PCR. We now no longer use the dandelions, which would be in lane 6. The dandelion smear is likely just contamination. Spinach is still 1,000 base pairs from before, so it hasn't really changed since then. And now onto the digest gel, which is right here. The PJET 1.2 plasmid is at work here, so the mini preps containing the BGL2 restriction enzyme are also at work, which are being cloned into the E-Coli. So you can see the strength of the first band here. The first band is here and here. And for mini preps 1 through 4, the bands are faint to see, so it's pretty hard to see, but the bands are definitely there. You can see 4 slight bands there, and on other groups there's also 4 slight bands. And these are actually 3,000 base pairs long, which is pretty significant. So figure 7 is the ligation transformation, and then figures 9 produced only 2 colonies, but that's fine. That's fine. So once we sent it off to the lab, they sequenced it. So here is the contiguous sequence, or the FASTA alignment view of the Arabidopsis thaliana GAPDH gene. So scrolling down, if I can get this to work, the blast results show a close homology between my cloned GAPC genes and the GAPC genes of chili peppers, pumpkins, and sage herbs, lemons, and Arabidopsis. So this is incredible because it shows the conservation of the GAPDH gene between very different plants. I can actually pull up this homology table right here. So the Arabidopsis was a 99.3% pairwise identity, 99.8% curia coverage, and 99.5% grade. It's very good. The sage, or Salvia elegans, 99.6% pairwise identity, 99.7% coverage, 99.8% grade. It's also incredibly good. Actually, that would probably be the best. The sage was the closest one, I'd say, other than Arabidopsis. Actually, no, in fact, it is better than Arabidopsis. The chili peppers, or Capsicum annuum, are 99.5% the same, with 99.6% grade. So actually, you're starting to see a trend here of these 3. They're all 99.6% grade, so they're very good, and they're all very similar. What that means is that these spinach glyceraldehyde 3-phosphate genes are actually exactly the same between chili peppers, pumpkins, and lemons. And the fact that it's the same between a chili, a lemon, and a pumpkin, and spinach, these are very, very different plants. Like, they're not similar at all. They diverged evolutionarily a long time ago. But because they were preserved, it actually shows that it's a set of genes used for glycolysis, catalyzing the 6th step of glycolysis. So this breaks down glucose for cell energy. These genes are so necessary for life that most organisms would actually die if they mutated. So when it comes to the Arabidopsis, I made a pretty good discovery here. On our homology table, it was expected that the end of the Arabidopsis gene, it would be located at this base pair, okay? 1,083,000. But mine was at 1,104,000 at the end. But I'm not the only one, so it wasn't an accident. There were other groups that found this 1,103,000 to 1,104,000. So it wasn't an issue with my cloning. This is actually a gene that within the Arabidopsis, it shows homology between my cloned cells and other people's cloned cells. But yet, when it comes to Genius actually did not recognize that, which I can show right here. If you pull up Genius, when these, see, these are the base pairs, of course. These base pairs, you can see that where it shows yellow, I had to modify those codes to make them more accurate. And that's by highlighting disagreements. So very difficult software to use, but if you zoom out, it starts to look like this. Where it shows red, it's a vector or a strong hit, so that's all removed. And you can actually easily remove most of the disagreements just by moving across these red bars. And that makes your sequences much higher quality. So actually, my sequences, when using Genius, created a 99.8% high quality, which is some of the best. I'm not saying I'm the best, but it's some of the best I've seen, and it's very good, at least for this experiment. I was not expecting it to be 99.8% high quality. However, if you move this over, just give me a moment, you can check the contiguous nucleotide collection, okay? So this shows in the contiguous sequence all these different plants. So if you go back to this, if you remember these where we have the, hold on, actually that's the wrong graph. Arabidopsis, sage, chili peppers, pumpkins, and lemons, that all comes from this. So these are different plants that have the same homologous structure for their GAPDH sequences. And they're all almost exactly the same, which is incredible to see that far separated animals, plants, basically all organisms, as long as they're eukaryotic, they share this same glycolysis genome. And the fact that almost all of these have grades between 99.5% and 99.7%, I mean, they're incredibly similar. It's almost mind-boggling to think about. And these graphs go on and on and on. Like, they just continue. It's very, very beautiful to watch sometimes. But if you go back to my assembly, and then you zoom in, it's amazing how we can use this new software from Bio-Rad to sequence the genome in a very cost-effective way. So the BLAST results show what I just showed you, because the BLAST goes through and separates all of the different the BLAST will use AI, and the AI will separate the genes and find ones that are similar. So in my acknowledgements, of course, thank you to Christopher Bayer and Western Carolina University Department of Biology and Chemistry. My references come from the Bayer and Harris Cell and Molecular Biology Manual. And of course over here, the Bio-Rad website, which Bio-Rad, it makes incredibly good the test kits you can create for yourself to do things just like this. So, in the conclusion and future work, the cloning of the GAPC gene from spinach to E. coli plasmid was successful, with just the usual human error from primer-dimers contamination on some PCR smears. But that's fine. It's almost impossible to remove all contamination. But the good thing is the Biorad's Genius software can actually cut out all that contamination. That's why I got a 99.8% accuracy. So you can actually use the software to sort of perfect it. And with future work, we can use this same exact software to software at least very similar to Genius to sequence the human genome. And for example, where I work, Pfizer, if you this is actually my dad's name tag, but at Pfizer, where he was the President's Circle corporate member, we have now created a 90 to 94% effective coronavirus vaccine. And the way that we have created a 94% effective coronavirus vaccine is simply by measuring the genome of the SARS-CoV-2 virus and measuring the human genome using this exact same software. And then we make a few edits like this in the yellow, and then we clone these. But instead of cloning it into a plasmid vector like we did here, we instead cloned it into a virus. And we have created an mRNA vaccine, which has actually never been done before. The coronavirus vaccine is the first mRNA vaccine. Because, as we know previously, most vaccines, you would kill a virus by denaturing it with heat and then inject yourself with the antibodies, and then your own body will create its own antibodies. We're no longer doing that. This is very experimental, but now we are able to create modified mRNA strands using this same exact software and modify human DNA. Ironically, it's actually illegal to modify the human DNA, but hey, with the new the government we have set up, I guess they just don't care anymore, do they? I mean, they'd rather create a cure, which I can understand, but keep in mind, it is risky and it needs more testing, okay? I'm telling you, it definitely needs way, way more testing, because you can't create an experimental vaccine in just 1 year and expect it to work all. So hopefully we'll get these mRNA-style vaccines up and running, which can actually cure all future diseases. All, literally, I mean every genetic disease, bacterial disease, and viral disease will soon be cured using mRNA vaccines. So that's why I wrote that this will help us use CRISPR-style gene editing, being CRISPR-Cas9, and cloning to create new vaccines and GMOs. So that's about it for my presentation here. So, hope you enjoyed this. It took many weeks. I think that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, around 10 weeks or so. So, have a good day.